Hi guys. Today we are going to go over 6.5 in the textbook, which is all about neurons and synapses. This is super interesting stuff. It's about how your nervous system works, but it is a fair amount of information. So let's get started. I'll let you read over the understanding, skills, and applications on your own. There's 3 slides of that, and then we go into part 1, the nervous system and neuron structure. So your nervous system is divided into 2 parts. Your central nervous system is your brain and your spinal cord, and your peripheral nervous system is your sensory neurons and your motor neurons. So your brain and your spinal cord are like the main show in the nervous system. They're what process the signals that are brought to them by your sensory neurons. Your central nervous system is the control center of the body, and its function is to interpret and respond to information from within the body and to the outside environment. So, your peripheral nervous system, their job is to bring that information to the central nervous system. So, you have sensory neurons that send informations from your sense organs, like your skin and your eyes, to your central nervous system. CNS is central nervous system. And then you have motor neurons that send information from the central nervous system to your muscles and other organs. So basically, you have one set of neurons responsible for bringing the information into the central nervous system. The central nervous system makes some kind of a decision about how to respond, and then you have another set of neurons that are responsible for bringing that decision out to your body so that your body can react. These things acting together, your peripheral nervous system and your central nervous system, we call that a reflex arc. So you have some kind of a stimulus in your environment. It might be like you feel a bug on your arm, and that the receptor cells in your skin travel that push that impulse through sensory neurons to your central nervous system. Your central nervous system, in that case your brain, and probably your brain. It could be just like a reflex that would be processed by the spinal cord. But in any case, some part of your central nervous system makes a decision that what you should do is brush that bug off of your arm. It sends that signal through your motor neurons to the muscles in your hand, allowing you to react and create a response to that stimulus. All right, so let's talk a little bit more about these neurons that are responsible for carrying these signals through your body. Your neurons have 3 basic main parts. They are the cell body, which is this whole thing in purple right here. The dendrites, which are these receiver areas on the cell body. And then the axon, and the axon is where a nerve impulse travels through a neuron. You'll notice the shape of these cells is pretty specialized to its function. You have an area where an impulse can be received, and that's a little bit wider, and then a super long skinny area where that impulse can travel. And the reason for the shape is we want that impulse to be able to travel as fast as possible through the axon so that you can react to stuff quickly. These axon terminal transmitters are usually pushed up next to the dendrite of another nerve cell, and that allows the signal to keep going through your nervous system in that reflex arc. We can go a little bit more into depth on the neuron structure. You have the nucleus, which is, like always, the control center of the cell. It transmits nerve messages through the cell body. And creates an impulse that then travels down the axon. The axon is covered by these cells called Schwann cells that together form a myelin sheath. And that myelin sheath acts as like an insulation on your neuron to allow that signal from the nucleus to travel down the axon as fast as possible. In this next picture, you have the axon and the Schwann cells pointed out a little bit more directly. So all of the Schwann cells together form this thing called the myelin sheath over the whole axon, but individually, each one of these little circles is a Schwann cell. and they insulate the axon of the nerve or of the neuron in a myelinated neuron. And between the little Schwann cells are areas called the nodes of Ranvier. And that basically allows for ions to diffuse out as needed from these little areas of the axon. And your nerves actually have this really cool adaptation where the impulse that you're sending down an axon is able to just jump over these nodes so that it can move even faster along the axon. The whole point of this is to get to the axon terminal as fast as possible, where you have this impulse transmitted to the next neuron structure in the line. And we'll go over a little bit more how that happens sort of at the end of this presentation. But you have the impulse leaving this terminal and then arriving at a dendrite. And that dendrite gathers information from the cell that's before it, passes that information into the cell body, and then the cell body figures out what to do. Like, do they need to keep the impulse moving down the nerve, or do they need to do something else? So, as we said when we were going over the parts of the neuron, most of your nerve cells are myelinated or insulated. And that myelin sheath exists to allow nerve impulses to speed up and move faster down the axon. So you can have those impulses jump these nodes. That's called saltatory conduction, from the Latin word for to jump or to leap. And nerve impulses can travel super fast through the axon, jumping over all of these little nodes. And like we said, the nodes of Ranvier, the IB syllabus doesn't really go into this, but it's important to have these gaps so that ions can diffuse in and out of the axon as needed to allow this chemical signal to move as fast as possible along the axon. And if you want to get a little sneak peek of how your nervous system works, you can take a paper clip and bend it into a U shape so that you have the tips separated by just a little bit. And try to touch this to different places on your body. If you touch it to your finger or to the inside of your arm or to your elbow or to the back of your shoulder, you might notice that it feels a little bit different depending on where you place the paperclip on your body. And that's because depending on where you are in your body, you have different amounts of nerve cells at the base of your skin. So like in your hands, you have a lot because you're touching stuff with your hands all the time. Your hands are super sensitive. The back of your shoulder, when you touch that paper clip onto that skin, it might just feel like there's one point on the paper clip. And that's because you don't have as many nerve cells reaching up to the skin there, because it's not that important to be able to feel really fine neural impulses in the skin on the back of your shoulder. So you can go ahead and give that a try, and it's just sort of like a fun activity to think about how your nervous system is processing the sensory information in your environment. And this slide goes a little bit over the information about why you get that feeling of like 1 versus 2 pinpoints when you do that. And the idea that the receptive field is larger in some places and smaller in others. Like you would say you have a large receptive field in your fingers because you have a lot of nerve endings in your fingers. All right. So let's talk a little bit about these impulses and how they travel along. If you want to pause this and watch the crash course video, I think this is queued up to run from minute 6 to minute 10, which talks a little bit about how a nerve impulse moves along your nervous system. And that's what we're going to discuss in this part of the PowerPoint. So nerve impulses can only travel in one direction. They go from the dendrite end of a neuron along the axon to the axon terminus end of a neuron. And they move through what we call local currents. So You have an electric current that exists in your nerve cells, and that current creates a potential. Basically, the idea that like a positive and a negative charge are separated by a concentration gradient create the potential for those charges to switch and do work as they're switching. And that's exactly what happens when nerve impulses get passed along your neurons. And to understand this a little bit better, we're going to talk about what your nerve cells look like when they're at rest, and then what they look like when nerve signals start traveling along them. So we'll start with what we call resting potential. And resting potential is the electrical potential across the plasma membrane of a cell that is not conducting a nerve impulse. So you'll notice that there's this imbalance of positive and negative charge. The outside of the nerve cell, this pink marshmallowy looking thing is your nerve cell. The outside is more positive and the inside is more negative. And we'll talk about why that is in just one second. But the idea that this separation of charges exists is what creates a potential for them to switch places and in doing so accomplish some kind of work along your nerve cell. All right, so let's talk about why this separation of charges exists and how it exists. You have in the membranes of your nerve cells active transport pumps that are sodium potassium pumps. And your sodium potassium pumps move ions to create this concentration gradient on the two sides of your nerve cells. You might remember that for active transport, you're using ATP. So if you can envision this as an active transport pump, a little ATP molecule comes along and hooks itself right in there. You can think of this like a little trap door that will swing open. So the ATP comes along, it's open on this side. 3 sodium ions come and line up. The ATP swings the door shut. The 3 sodium ions get pushed to the outside. 2 potassium ions come along and join up on the other side. And the door swings the other way and allows the potassium ions to come inside. You'll notice that both of these ions are positively charged. So it's a little bit weird to think that overall this creates a concentration gradient where, I'm just going to go back for 1 second, the outside is more positive than the inside. But since 3 sodium ions are moving out and only 2 potassium ions are moving in, you do end up with a more positive charge on the outside of those neurons. That is important because it creates this resting, I'm going to go back again, it creates this resting potential where you have a positive charge on the outside and a negative charge on the inside. Then, when some kind of a stimulus comes along, you can do work, you can create an action potential by switching the charges. So an action potential is a nerve impulse transmission down a neuron, and it's how electrical signals travel along the neuron after a stimulus. It involves the depolarization and repolarization of the inside of the neuron. So you can see here, like in a normal time at a resting potential, the inside of that neuron has a voltage charge of about negative 70 millivolts. When it gets depolarized, when it changes due to some kind of a stimulus, it shoots all the way up to about positive 40 millivolts. And then once the stimulus has moved along the neuron, it goes back down and actually overshoots its target potential a little bit to ensure that the signal can't like move back up the neuron at the wrong direction. But then eventually it goes back to its resting potential, its resting state of about negative 70 millivolts. And this is, we're talking about inside of the neuron cell. So we're looking at this like the depolarization, the switch that happens inside of the neuron from a negative charge to a more positive charge and then back again. And we call this kind of drawing an oscilloscope. And we'll talk about it a little bit more in some more detailed steps over the next few slides. So let's start at the very beginning. We have some kind of a stimulus applied to our neuron. It could be like a chemical thing, an electrical thing, a thermal thing, or a mechanical thing, but it does something that causes a sodium ion channel to open. And sodium ions, positively charged sodium ions, start to diffuse into the neuron. And it causes the membrane potential to rise from being really negative to being a little bit more positive. Because remember, these ions are positively charged. So in order to actually do anything to transmit the signal successfully along the neuron, this stimulus has to exceed what we call a threshold potential of negative 50 millivolts. So as long as the stimulus causes the sodium channels to open enough to allow enough sodium ions in, that they'll increase the charge of the inside of the neuron by about 20 millivolts. We're good to go, and then you trigger like a self-propagating action potential where more sodium channels open and the nerve is going to transmit the impulse along the axon. So we have our stimulus applied. Once we start opening more sodium gates, we move into this stage called depolarization. And remember, that's when the charge inside of the neuron switches from being positive to, from being, let's go back here, from being negative to being more positive as more sodium ions are rushing in to the neuron. So we're here, depolarization. Sodium ion channels are open, and there is a huge influx of sodium ions into the neuron. So at that point, sodium ions are just diffusing into the neuron because we've done all of this active transport to like pump them all outside of the neuron. Once these channels open, they want to just flow from high concentration to low concentration to the inside of the neuron. And that obviously creates a more positive charge inside of the neuron because all of these ions have a positive charge. The potassium gates are closed, so the potassium ions are all stuck in there also. And you get the positive charge that shoots up to about plus 40 inside of the neuron. At this point, we enter a phase of this nerve impulse called repolarization. And that is when the neuron wants to write the charge potential on each side. So at about plus 40 millivolts, what we call peak potential is reached, and the neuron sends a signal to close those sodium channels so that no more sodium ions can rush into the cell. It sends another signal to open the potassium channels, and potassium starts flowing out of the neuron super fast. So this restores the inside negative charge resting potential of negative 70 millivolts, and really even like a little bit, it ends up going a little bit past that. And when it goes past it, we call that a point of hyperpolarization. And this is what ensures that that signal can only move one way down the axon because the potassium channels remain open and potassium ions keep flooding out of the membrane and trigger the next part of the axon to have that same action potential go through it. So it ensures that things are only moving one way. And once you get lower than negative 70 millivolts inside of the neuron, eventually the potassium gates close and the sodium potassium pump turns back on because remember, we have the charges right now but the ions are in the wrong place. So we don't just want to have the inside be negative, we want to have the inside be negative and full of potassium ions. So the sodium-potassium pump restores the resting potential and puts all of the sodium ions outside of the neuron and the potassium ions inside of the neuron. There's a time where the membrane can't handle another stimulus when it's still like righting itself during this time of hyperpolarization, and we call that the refractory period. That's not like super important on the IB syllabus, but just for your knowledge. So on a graph like this on an oscilloscope, you should be able to label depolarization, repolarization, and hyperpolarization. You should also be able to show when sodium ions are moving in, when potassium ions are moving out, and when the sodium-potassium pump is building up the gradient. So let's talk about each of those things. Remember, resting potential, we're down here at negative 70 millivolts. Depolarization, and this is, remember, inside of the neuron, so negative 70 millivolts inside of the neuron. Depolarization is when that area inside of the neuron switches from negative to positive. So depolarization is here. Repolarization is when it switches back. So repolarization is here. And hyperpolarization is when not only does it switch back, but it goes even lower than it's supposed to ensure that one-way movement. So hyperpolarization is here. The sodium-potassium pump turns on right around here to restore the normal resting potential to the nerve cell. So over the next couple slides, all of these things are correctly labeled. You can check the work in your packet. And we can talk a little bit about what happens when the nerve impulse gets to the end of one neuron and it hits something called the synapse. So synaptic transmission is how a nerve impulse is passed from neuron to neuron. So there's this little space between neurons that's called a synapse where the neurons aren't actually touching each other. And we need to have some way for signals to move across this like empty space. We need to make some kind of a bridge that allows nerve signals to move from what we call the presynaptic neuron, the first neuron, to the postsynaptic neuron, the second neuron, the neuron after the synapse. And we call this area, this this fluid-filled gap between the plasma membranes, we call it the synaptic cleft because there's like a little cleft, a little break there between the two neurons. And in a chemical synapse, electrical impulses can't cross the gap. So you do have in your body electrical synapses where like the signal needs to move super fast and it just like an electrical signal can just like leap across there. But what the IB syllabus mostly focuses on is these chemical synapses where electrical impulses can't cross this area and they need to come up with some other way to get the information from the end of one neuron to the beginning, the dendrite of the next neuron. And in a chemical synapse, we call it a chemical synapse because in this gap we use chemicals. And those chemicals are called neurotransmitters. Your body stores them in synaptic vesicles, so like these little circles are full of neurotransmitters, and when a nerve impulse reaches the end of the axon and comes up to the axon terminal, all this fancy stuff happens to allow the vesicles to move across the gap. And we will talk about what all that fancy stuff is right now. So there's a little video that outlines this movement if you want to pause this video and go ahead and watch it on the PowerPoint, and then I will talk you through each step of this. So synaptic transmission, we're going to start with this overall like diagram of everything that's happening in synaptic transmission. The first thing you have is this nerve impulse moving to the axon terminal of one neuron. And as that nerve impulse moves into the axon terminal, the charge, the electric charge, we're still electric here because we haven't left the neuron yet. So the electric charge causes the opening of calcium channels in this part of the neuron. And those calcium channels allow calcium ions to come into the axon terminal. And those calcium ions bond to these synaptic vesicles. So these little circles, or bubbles, I guess is a better word, full of these neurotransmitters. When calcium ions bond to them, they sort of drag them over to this area at the very end of the neuron, and through exocytosis, the vesicles leave the end of one neuron and dump all their neurotransmitters out into the synaptic cleft. The neurotransmitters are then picked up by the dendrites on the next neuron. And we have all of this written out in individual steps. So let's read through them. The first thing that happens is the action potential reaches the terminal end of the presynaptic neuron. That action potential causes calcium protein channels to open and allow calcium ions to enter into the end of the neuron. Those calcium ions move into the presynaptic neuron via facilitated diffusion because they're in these protein channels. Next, vesicles containing neurotransmitters fuse with the plasma membrane and release their neurotransmitters. And remember, we call that exocytosis. Neurotransmitters diffuse across the synaptic cleft, and neurotransmitters bind with a membrane protein on the postsynaptic neuron. And that binding can actually cause like a bunch of different things to happen, because you can have excitatory or suppressatory neurotransmitters. But in this case, let's just say that the binding causes sodium ion channels to open on the postsynaptic neuron. Sodium enters the postsynaptic neuron via facilitated diffusion. It starts an action potential, which will then self-propagate down the axon. And once that action potential is safely passed on to the next neuron, the neurotransmitters are either dissolved by enzymes in the synaptic cleft, and presynaptic neuron can make new ones, or they're pumped back into the presynaptic neuron via active transport. So sometimes, I don't know if there's one in this picture, there's not, but sometimes like right here, you would have what's called a reuptake channel, where the little neurotransmitters can like choo 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 come over here and get pumped back into this end of the presynaptic neuron and get rebundled into vesicles and do the whole thing again. And those are the slightly more detailed steps of synaptic transmission. There are a couple of specifics about synaptic transmission that the IB syllabus goes into for applications. One type of neurotransmitter that you should probably know by name is acetylcholine. And acetylcholine is a neurotransmitter that works between neurons and muscle cells. It's not super important that you understand like what it's made of, but it's kind of interesting. So you get choline from your diet, it's a form of vitamin B, and you get an acetyl group from aerobic respiration. You might remember in the Krebs cycle we talked about like coenzyme A and acetyl groups. When coenzyme A and the acetyl groups are done in the mitochondria, they can come down into the presynaptic areas of your nerve cells. So you have these two things bond together, and then when they're bonded together, they function as a neurotransmitter that gets sent across the synaptic cleft and then gets used to activate like a muscle response. And there's a pretty good picture of that here. So you have acetyl-CoA and this choline molecule. Those two get bonded together with an enzyme to form acetylcholine. That acetylcholine gets shoved into a vesicle. The vesicle, through exocytosis, goes into the synaptic cleft, and then you can see like this is its little receptor. And in your nerve cells, there's all kinds of different receptors for all kinds of different things. So this one is specific to this type of a molecule. This neurotransmitter bonds to the receptor for long enough to pass the signal along to your muscle cells that your muscle cells need to move somehow to react to a stimulus. And then this molecule, acetylcholine, is broken down into its component parts by an enzyme. So the reason that I don't really know the reason why the IB syllabus asks about this, but it's connected to another application that they ask about, which is a type of pesticide called a neonicotinoid. And what neonicotinoids do are they bind to these acetylcholine receptors in the postsynaptic neuron and block the receptor so that acetylcholine can't bind. So there's no nerve transmission. And remember, these are the things that are in control of moving your muscles. So if the acetylcholine can't bind, the insect gets paralyzed, and eventually that receptor gets overstimulated because there's this thing like binded, I think you say binded to it that won't move. Bound, I think it's bound to it. The receptor gets overstimulated, and the insect can't receive any other nerve transmissions. The insect gets paralyzed, and eventually the insect dies. And there is some, like at first, people and insects have slightly different acetylcholine receptors. So at first, people thought that this was like a great insecticide because it didn't cause like really specific, obvious harm to human beings, but it killed insects. And now, as we've continued to use these insecticides, there is rising controversy. People think that this type of an insecticide might be responsible for colony collapse disorder, the problem that's killing honeybees. And people are also finding out that like there is some harm to human beings, not surprisingly, of like having this toxic chemical building up in our environment. Especially like in fetal development, they're finding that there might be some really scary ties about like fetal brain development and the presence of neonicotinoid insecticides in the environment. So you can see in this diagram how these neonicotinoids work a little bit better. Like in normal functioning, you have acetylcholine being formed in the transmitting neuron, the presynaptic neuron, going into the cleft, binding on to these receptor proteins in the receiving neuron, the postsynaptic neuron. Eventually, it transmits the signal it needs to transmit, gets broken apart by this enzyme, and then the component parts can go back into the transmitting neuron, and that can just happen as a cycle. When you have these neonicotinoids, instead of the acetylcholine bonding to the receptor proteins, the neonicotinoids bond, and you have this enzyme that is not capable of breaking down neonicotinoids, so they stay in these receptors and block everything else that is supposed to be entering those receptors. All right. Last thing, and this is just for your interest, drugs affect nerve transmission, and that is why they make you feel different ways. So they alter usually the release of a neurotransmitter or block a receptor that a neurotransmitter is supposed to go to. So that you have either like a rush of neurotransmitters or neurotransmitters that are not effectively taken up, and those affect your mood. And that is how drugs work. And let's see. There's an IB practice question on here about explaining the events that occur during a nerve impulse and how the impulse is propagated along the axon. So remember, you don't necessarily have to talk about synaptic transmission here, just like what's actually going on in the neuron. You can check your work. I'll skip over that slide here so that I don't spoil the surprise. There are extra resources and animations if you want to review any of this a little bit more and read over 6.5. This is, I'll say again, a lot of information. So if you have any questions, we'll have time to go over those when we are all back together, which I think will be next week. All right, so that does it for now for 6.5 about neurons and synapses.